Um, Hi, good evening. Thank you for joining. Am I audible and is the screen visible to you? Is my screen visible? Okay, let me know if you can see my slides whenever you are seeing the slides. If you can't, let me know. I will share it again. Okay, so thank you for joining. This is problem solving session 11. The second last session where we are covering the topics of the course. There will be one more session, which will be the last session for covering theoretical classes and all the topics that you've studied so far in this course. Um, we are trying to put a 13th session, which would probably be on 16th, which is next Wednesday, not the next Tuesday. So next Tuesday, which is 15th, we'll have a regular class, like we will discuss the course for. And on 16th, we're trying to put the last problem solving session, wherein, as I have mentioned in the previous weeks, also that we will briefly go over all the core syllabus. But what I would do, in case if you don't have any particular pressing questions, then I would have questions from all these different topics randomly, like mixed and matched, and also have several numericals, wherein the questions will be from the assignments given this year to you with their solutions. And you would have already received these solutions for the assignment questions of this year, so that is also there with you. But what I would do is pick out some questions and discuss them. In case you have any trouble with any other question, you can let me know. Those questions will be there on the slide. And we'll do numericals like we have done in the first few sessions. We'll just have a brush up of all those. Okay. All right. So with that, if they are trying to put that session, let's see if that turns out to be a schedule, then we'll get to know. Now coming to today's session, like last time we had discussed about anabolism and catabolism of our biomolecules. Today also we will be discussing about similar pathways. This time the biomolecule is fatty acids. So we'll first look at fatty catabolism, which is breaking, how are fatty acids broken down and how do we get energy from them. And the second half of today's session will be about how are fatty acids synthesized. So if you're breaking down, you're also synthesizing them. Today also, I have many questions that can help you, later that can serve as a revision questions for you whenever you're trying to revise these topics, right? Okay, with that background, let me begin. Okay, so fatty acid, the term, it is basically triacyl glycerol that we already know. That's what we are consuming. So the monomeric units of TAGs or TAGs are to call fatty acid. Now the oxidation of these long chain fatty acids result in acetyl coenzyme A, and we know acetyl coenzyme A is a central energy yielding component. So overall, the oxidation of long chain fatty acids to acetyl coenzyme A is a central energy yielding pathway in many organisms and tissues. And you would see that in mammalian heart and liver, it provides as much as 80% of the energy needs for all physiological circumstances. So the very important energy yielding pathway. We already heard about glucose pathway. We have seen how amino acid degradation takes place, but we'll see now how fatty acid degradation takes place, and that's almost contributing to 80% of the energy requirement, right? And similar to the other two pathways, the electrons that are removed from fatty acids during these oxidation steps, they pass through the ETC or the respiratory chain, and they drive the ATP synthesis. While the acetyl coenzyme A, which is produced by the oxidation of fatty acid, that, as we know, as we have already discussed this, it is completely oxidized to carbon dioxide in the citric acid cycle. So we have oxidized carbon dioxide and we have ATP synthesis, which is a result of oxidation of long chain fatty acids. Now, the oxidation of fatty acids is called the beta oxidation, and it has the central four processes. we'll see Y beta oxidation as well, right? Now, before we go into how we are oxidizing, we'll first see how do we access fatty acids, right? Now cells can derive these fatty acids in three ways. One is fat, which is consumed in diet that we are quite well aware of. Second is fat, which is stored in the cell as lipid droplets. Remember, we are talking about breaking, right? So there is also a form which is stored. So in the stored form also, it can be used when it is mobilized from those cells. And also fats which are synthesized in one organ for export to another on the way, or also it can be utilized for source as an energy. So you have dietary lipids or dietary fats. Then you have fats which are in adipocytes, and then fats which are synthesized in one organ for transport to other, right? Now, we will first see how the fats are something that we are more commonly doing. We are consuming fat. So how that consumed fat which is consumed, how is that being used up, right? How is that uh, giving us the fatty acids? Okay. So this here, this figure here that you see, it's basically telling you the process of how dietary lipids are utilized, right? So as you can see in the beginning, you have the fats which is ingested. Just a second. Yeah. So your fats are ingested and it enters your stomach. Then it goes to your small intestine. Now, small intestine is where your absorption of fatty acid takes place, right? So first, what is the first step that takes place? You have the bile salts here, which emulsify the dietary fats in the diet. So they emulsify the fats here, the bile salts, and they form something called micelles, right? So the micelles are basically emulsification of the lipids or the fats. Then what happens next is that there are lipases. Lipases break down lipids. So there are intestinal lipases. They will start breaking the TAGs or the triacylglycerol, which are the structural component of lipids or fats. Right, so they start breaking this. Now the third, after this, what happens is it's fatty acids and other breakdown products. So I told you, when you break down TAGs, you get the fatty acids. If you remember the structure of triacylglycerol, glycerol backbone, you have three acyl groups. Those are the fatty acids. So when you break down or when you disrupt the structure of lipid, you get fatty acids and a glycerol backbone, right? So this fatty acid that have come up as a breakdown product, they are taken up by the intestinal mucosa and they are converted again into THs. Okay, breaking down and taking them again. What happens after this? So these THs are now incorporated with cholesterol and apolipoproteins into chylomicrons. So these THs, which are now again in the intestine, and you have cholesterols also, which are part of the dietary component lipid that we have taken. They're part of chylomicrons. These chylomicrons, the chylomicrons will move through the lymphatic system and bloodstream. We know this. They travels via blood. Then we have lipoprotein lipase. So remember, it's a lipoprotein, right? So you have lipoprotein lipase to break down this component. So this lipoprotein lipase, which is activated by ApoC2 in the capillary here, blood capillary, this converts the TAG again back to fatty acid and glycerol, right? So the actual action is after it has formed chylomicron, you have lipoprotein lipase, which is actually breaking down to fatty acid. And that fatty acid can enter these cells, right? Because this fatty acid, so the exchange has to be between the blood and the cell. But for it to come into the blood, it has to pass through the small intestine, right? And when it enters the blood, that's when they are oxidized or re-acidified for storage. It can be adipocyte for storage, or it can be liver where it can be oxidized or where it can be utilized. So you have these different outcomes, but the food that we ingest has all these different steps going on till it finally becomes fatty acids, which can enter the cells, which can enter the cell for further oxidation, right? Okay. Now, this I told you about one way of cell to access or utilize fats was diet. The other was, if you remember, I told you about the ones which are stored in the tissues, stored in the organs. Those stored. Hello.
Second article is
Okay. Sorry. I think there was just Yeah, coming back to it. So what I meant was that one way the cell can access your fats or fatty acids are by diet. The other is when they are mobilized or when they are moved from the storage form. Where they are stored in adipocytes. So when you mobilize your adipocytes lipids, that's also a way that the cells can access fatty acid. Now what happens in those, what access or what helps in mobilizing these stored fats from adipose tissue, you have hormones, right? So this is the way this on your slide shows you how it's mobilizing. Now in this part of your course, you will have a lot of, as we have already seen, each pathway is very important and each component is very important. You can there's a general understanding of the regulation. You know what's regulation, how can it happen, you can understand it, but you particularly need to be careful about the enzymes and the components that are part of it, the precursors and where they are taking place, right? So when you are studying or when you are revising, just be more mindful towards this. Okay, so there are a lot of names, but if you carefully read it, I'm pretty sure it's not that difficult to understand. All right. So now, how does mobilization of fatty acids take place, right? So these are neutral lipids which are stored in adipocytes and in the form of lipid droplet, as you can see here. So these are the lipid droplets. So this is where you have your fats or fatty acids stored, right? Now, this lipid droplets or lipids, as we know, they have a core of sterol esters and TAG. So these are surrounded by phospholipids, as you can see on the slides as well, right? Now, what you need to remember, the surface of these droplets have something called perilipin. Okay, remember this name. The name is very simple. Peri means surround, and they are surrounding lipids, so they are perilipins. Lipins means a molecular protein or a family of protein. So perilipins. So these are family of proteins who are on the surface of these lipid droplets. And what do? They restrict the access of to lipid droplets. You don't uh, any other component in the cell cannot interact with this lipid droplet, and it therefore prevents the lipid mobilization, right? So basically it's like this lipid is here, a lipid is there. For you for cell to mobilize this lipid out of this droplet, these lipids need to be removed, right? So um, when there is hormone signal, this removal is under the influence of hormone. So when there is hormone signal for the need of metabolic energy, these TAGs which are stored in the adipose tissue, they are mobilized, they are brought out of the storage as we have discussed, and then they are transported to the tissue, be it skeletal muscle, be it heart, be it renal cortex, wherever, in which they will form the fatty acids, and the fatty acids will be oxidized there for the energy production, right? Now, this whole cascade depends on the cell getting removing the perilipin, but it has to be under very, very strong regulation. Otherwise, if it is easily mobilized or if it's easily removable, then there's no point of perilipin being on the surface of the lipid droplet. So you will see that this removal of perilipin or this moving out of from the storage of the lipid droplet is again a 2-3 step pathway, right? So first of all, as you can see on the slide, the hormones which are responsible for mobilizing these fats are epinephrine and glucagon. Okay. These are secreted in response to low blood glucose level. Now, if you have low blood glucose level, you need more energy because you don't have energy. So these hormones, epinephrine and glucagon, they signal the body to release more energy. One of the way that they will interact with the cell is to say that, okay, release more of the nutrients, release more of the source, and the source will be adipocytes. So the adipocytes will release these lipids which are stored. So under the influence of glucagon and epinephrine, this mobilization will take place. Now, how will that take place? The first thing that these uh, hormones will do after binding to its receptor is it activate the enzyme adenylyl cyclase, right? It's in the plasma membrane, as you can see here. It will, via G receptor, it will bind and it will activate this adenylyl cyclase. Now, what will adenylyl cyclase name suggest to you? That it will produce something called cyclic AMP, CAMP. This cyclic AMP then will further go to a protein called protein kinase A, which is here. Protein kinase A is dependent on this CAMP. So whenever it detects a CAMP, what will happen is that this will uh, influence or induce the protein kinase A to go and phosphorylate the perilipin, add a phosphate group on these perilipins, right? So cyclic AMP dependent pKa phosphorylates the perilipin A, and the phosphorylated perilipin now, this cause hormone sensitive lipase, right? So this phosphorylated perilipin, they will cause the hormone sensitive lipase in the cytosol to move to the droplet surface, and there, this, as the name suggests, they are lipases, so they will come to the surface of the lipid droplet and they can hydrolyze the TAGs to free fatty acids and glycerol. Okay, this PK, not only the perilipin will induce these hormone sensitive lipase, the pKa, which is here, as you can see on the slide, pKa also is able to do the same. So pKa also phosphorylates hormone sensitive lipases, and because it phosphorylates, it makes it more active and its activity increases. So phosphorylation of your lipase makes it more active, so you can have more of this breakdown. So you can see the hormone goes to this adenylyl cyclase, CAMP goes to PK goes to perilipin, PK also does this hormone sensitive lipase phosphorylation, which makes it more active. The ultimate goal is to break down the lipids, make it into free fatty acid from. Now this is an adipocyte. This adipocyte has to go in, from the adipocyte the fatty acid has to move into the blood, and from the blood it will be then taken to wherever it has to go. All right. So now what happens? How do that happen? So whenever this breakdown happens, the free fatty acids, so whichever fatty acids are broken down from the stored form, they are called free fatty acids, or FFA. You will often see this name, right? So this FFA, they will pass from the adipocyte into the blood. Simple to understand. How are they carried in the blood? They are carried when they are bound by albumin. So these fatty acids, they are bound to albumin and they are transported in the blood, right? And then what happens is, when they reach the surface of the distant sources of Tostin cells like skeletal muscles or renal cortex, there their exchange will take place and they will enter. We will see how they are entered and we will see how that goes ahead. But this is what happens in general, right? Now, we are only talking about fatty acids. What happens to the glycerol backbone, glycerol? So as I said, that in TAG, you have a triacyl component and a glycerol component. But 95% of the available energy of these TAGs are in the fatty acids. That's why we are talking about fatty acids always. The glycerol that is there, that also forms when there is a breakdown of TAG, only contributes 5% energy. And the glycerol, what happens there is, when the glycerol is released, it is phosphorylated by an enzyme called glycerol kinase. And that glycerol kinase, as you can see here on the slide, this glycerol becomes glycerol kinase. So glycerol becomes L-glycerol triphosphate, which will become DHAP. And if we know DHAP, we know that we have already studied this in glycolysis, which is again a pathway for energy generation. So your glycerol is incorporated into the glycolysis, but that's only 5%. Majority comes from the fatty acids, the free fatty acids, right? So, yeah. So what we have seen is dietary components. If I go back to the previous slide, dietary components have entered the cells. If I am mobilizing the stored form of TAG, they are also now into the cell. Now, before I do that, before I go into the oxidation, we will first also look into the what is the step before your oxidation takes place. Okay, how do this oxidation actually takes place is what we're going to see now. Okay, now just a second. Yeah, so what we see here is fatty acid which has entered the cell. It undergoes a conversion to give rise to fatty acyl coenzyme A. Okay, Now, fatty acyl coenzyme A is important, as we will see that this um, when it is in the cell, this is what will lead to the oxidation. This oxidation of coenzyme A component is what is important, right? So first we'll just see that to understand that the fatty acid is converted to fatty acyl coenzyme A by these steps here. Okay, so if you see, basically to understand which enzymes are taking place here, this conversion to fatty acyl coenzyme A is basically carried by fatty acyl coenzyme A synthetase. Remember synthetase, right? So if it is a synthetase, it basically uses ATP, as we had already established, and you can see that ATP here, right? So your the acyl coenzyme A synthetase catalyzes formation of a thioester linkage between the fatty acid carboxyl group and the thiol group of the coenzyme A. That together makes it a fatty acyl coenzyme A. And as you saw, it requires the cleavage of ATP, right? And the delta G for this is minus 9 and minus 15, so they can take place in a it that's like it's spontaneous, it is favorable, and this is a two-step reaction. There's an intermediate form and then it's the final form. Okay, now why are we talking about fatty acyl coenzyme A? What is the importance here? So here, let's go to the next slide first and then try to understand this, right? So this enzymes of fatty acid oxidation in the cell, it is in the mitochondrial matrix. So the fatty acids now have to enter the mitochondrial matrix, right? Now, remember this, the fatty acids which have a chain length of 12 or fewer carbon, 12 CN or lesser than 12. These can enter mitochondria without the help of any transporter. They are small molecules, they can easily enter the mitochondrial matrix, right? But with the bigger ones, 14 or more, how will they enter? For them to enter, they since they cannot pass directly, they will use something called shuttle. Okay, so this shuttle will allow them to go inside the mitochondrial matrix. Now what is that shuttle? Shuttle is making, basically they're moving via a transporter, and that shuttle is called carnitine shuttle. And here, if you see, for that shuttle, the first reaction that takes place is by these family of isozymes here, uh, which are present. So now these enzymes are present in the outer mitochondrial membrane here. So if you see, they have to go here. They're here. For them, so if you remember, this is cytosol, this is the matrix, this is the intermembrane space of mitochondria. Mitochondria is a double layered membrane space. So there's internal space, outer, inner matrix. You have to locate yourself through all of these membrane to enter matrix. So in the cytosol, this fatty acid will become a fatty acyl coenzyme A, and this fatty acyl coenzyme A, which has already like utilized ATP, this will now enter it the mitochondrial matrix by this entire procedure. Now what happens? So you can see here, when you have a fatty acyl coenzyme A, there is an enzyme called carnitine acyl transferase. So that enables the fatty acid to bind to carnitine, and that carnitine can enter via the inner mitochondrial membrane. Okay, so to enter the mitochondrial matrix, you need the fatty acids to convert into fatty acyl coenzyme A. And the enzyme which is important here is carnitine acyl transferase, because carnitine is the transporter protein here. The carnitine protein is what will enable this movement of the fatty acids from the cytosol to the matrix. Okay, so yeah, after coming back, you have your fatty acyl coenzyme A back, right? Now we will see what happens. So it's easy, right? You have made fatty acyl coenzyme A. You have now bound to carnitine. With carnitine, you come inside, and then carnitine with carnitine removal, you have the fatty acyl coenzyme A inside. Right? Okay. Now what happens inside is fatty acid oxidation. So if you if you have to look into fatty acid oxidation, the first step is to get fatty acid inside the cell. So that itself has 2-3 steps that we just discussed, from fats to fatty acid, from fatty acid to fatty acyl FST acyl coenzyme A. Once it has come to that form, then it will undergo oxidation to what we call fatty acid oxidation, which itself again has 3 stages. First stage is a long chain fatty acid where it is oxidized to yield acetyl residues in the form of acetyl coenzyme A. It already has a coenzyme A. These will all get oxidized and form acetyl coenzyme A. Right? The second step, as we are all aware, the acetyl coenzyme A will be incorporated into the Krebs cycle to yield CO2. Third, as we have already discussed, the electrons which are part of these stages 1 and stages 2, they move to the ETC, and from there they are used for ATP synthesis. So stage 2 and stage 3 are clear to us. We have already discussed this multiple times. Let's look into the stage 1. What exactly happens in the beta oxidation here? How does it take place? Right, so we understood that all the beta oxidation can lead to acetyl coenzyme A. We look into this in more details now. Right, so fatty acid oxidation or the beta oxidation state of fatty acid has 4 basic steps. But remember, fatty acids, as we have discussed, they are saturated, they are unsaturated. Saturated means all single bonds, unsaturated means double bonds. Unsaturated can have polyunsaturated and monounsaturated, so mu-fuzz and pu-fuzz. So the way these are broken down, they are similar, but there are additional steps for the mu-fuzz and the pu-fuzz to bring them down to the common intermediate state. We'll see all of this. I'm just giving you a heads up that what we are discussing right now, the first 4 steps or the first breakdown, they are for saturated fatty acids, which means they only have one bond, single bonds. Right? Okay. Yes. So coming to the fatty acid oxidation, and we'll start with palmitoyl coenzyme A, which is a 16 carbon chain of palmitate. So we'll see how this oxidation takes place. Okay, how this, so if you just overall look into it, you will see a very clear pattern that, let's say we begin with C16. So C16 to C14 to C12 to C10 to C8, C6, all this. And all these breakdown is vertic. What you're seeing is 2 carbons are being removed as acetyl coenzyme. Okay, this is the these are the pink shaded ones. The ones which are colored in pink here are the ones, are the carbons that are removed, and they're removed as acetyl coenzyme A. So if you start from palmitate, which is a 16 carbon, at the first step you have acetyl coenzyme A removal. So 2 carbon is removed. So you have 14 carbon left. Again, next round 2 removed. So this way it will consecutively keep removing. You have 16 carbons, so you need 8 such cycles to remove it. Or to break it down to acetyl coenzyme A. So 16 carbon will give you 2 carbon acetyl coenzyme A, right? And how does one acetyl coenzyme or the first breakdown take place? By this 4 steps. The first enzyme is acyl coenzyme A dehydrogenase. Second is enoyl coenzyme A hydratase. Third is beta hydroxy acyl coenzyme A dehydrogenase. And fourth is acyl coenzyme A acetyl transferase. These are also called thiolase. Yes, it looks very heavy name, but that's how it is. And these are, if you read by, if you remember the components which are formed in each step, it should be intuitive to you, right? But if you look at overall, if for a formation of each acetyl coenzyme here, you can see there is removal of 4 hydrogen atoms, okay? Or there are 2 pair of electrons and 4 hydrogen atoms which are associated with the formation of acetyl coenzyme. This stoichiometry is what you need to remember. Okay. So there are 4 hydrogen removal, 2 pairs of electrons removal for one acetyl coenzyme A formation. One acetyl coenzyme A is basically one cycle of fatty acid oxidation. Palmitate, which is 16 carbon fatty acid, takes 8 cycle, but if it's a 14 carbon fatty acid or 12, it requires lesser. Okay, so what you need to remember is look at the number of the carbon and then just divide it by 2 to know how many cycles or how many acetyl coenzyme A will be synthesized or produced. Okay, right. So just let me quickly tell you what is happening in each step so that we just quickly do it. We don't want to go into more details here. So the first step here, which is your of fatty acid oxidation, you have the dehydrogenation of fatty acid coenzyme A. As you dehydrogenate it, if you see here, as you dehydrogenate this compound, it forms a two carbon double bond, right? So this hydrogen is removed and you have a double bond here. Now, because it's a double bond, it's called trans because they are in trans orientation. Delta square enoyl coenzyme A. Coenzyme A is still there and it's an enoyl. So you have trans delta square enoyl coenzyme A. Now the next enzyme is enoyl coenzyme A hydratase. So you're basically adding water. Now you are to same keep looking at keep tracking the pink one always, pink shaded area. Now as you add water, you have H O here and H1 here. So this becomes something called L beta hydroxyacyl coenzyme A. So the double bond is lost and you have a COH and H and CH2, right? In the next, again, you have a dehydrogenase of the same compound here. So beta hydroxyacyl coenzyme dehydrogenase. So dehydrogenation, hydration, and dehydrogenation. And this dehydrogen leads to this group here, CO, okay? And you already have a CO here. Now, if you have acyl coenzyme acetyl transferase, okay? So what it does is it's basically transferring your acetyl group. So you have a coenzyme A here. Now this addition of coenzyme A, this goes to this oxygen here. Sorry, yeah, this carbon here, and this part is broken. So one coenzyme is added here, and the other coenzyme A is on this component. So you have one acetyl coenzyme A, which is coming out from this part. Now the remaining, what is the remaining becoming? The remaining is becoming the 14 carbon compound with coenzyme A. So myristoyl coenzyme A. This is what is 14, right? So you have dehydrogenation, hydration, dehydrogenation, and acetyl transferase. As you have acetyl, you have one acetyl on this acetyl coenzyme and the other acetyl on the myristoyl coenzyme, which is the fatty acid with two less carbon. This is what is happening in fatty acid oxidation. And now in next round, this myristoyl coenzyme is basically what is here. This will have dehydrogenation. Again, the alpha and the beta carbon will form double bond. Now, why was it beta carb oxidation? Because this, if you see, is getting oxidized. So beta oxidation. That's what it's all about, right? Okay, so we have understood the basic. We can look at the stoichiometry as we have already discussed. If you look in one pass, palmitoyl has coenzyme A here, plus FAD and NAD, FAD here. And so the two dehydration step, one uses FAD, one uses NAD plus. So FAD and NAD plus H2O, the second step hydration, which will give you myristoyl coenzyme A, acetyl coenzyme A, one FADH2 and one NAD. So this, if you remember, you will remember the steps also, right? Now, if you look into the overall beta oxidation, there is 7 coenzyme A because this has to pass through 7 cycles, right? So this has happened once. This at the end, the 7th cycle, this will again give 2, right? So 7 such coenzyme A, 7, all of these 7, and this acetyl coenzyme will be 8. Because right now you have one acetyl coenzyme and something else. But in the last step, you will have 2 acetyl coenzyme A. So 8 acetyl coenzyme A. So when you join this and this to look at overall from the first step, you basically this can be your ultimate equation with ATP also in the accountable. What ATP? Why ATP? Because you are using ATP when you are synthesizing the acyl coenzyme A. So this is how your reaction overall oxidation looks like. Okay. Yes. Yeah. That's about ATP. Now remember this overall oxidation of palmitoyl. This is for palmitoyl coenzyme to 8 molecules. Okay. Now, in the second stage of oxidation of palmitoyl, it is also coupled with phosphorylation, right? Which is the third stage. So basically, what is happening here is this is stage 1. two stage 2 and two stage 3. So when you go to the stage 2, you have ATP. When you go to the stage 3, you have CO2. So if you look into the overall stoichiometry for stage 1, stage 2, stage 3, this is the overall for all the stages. This is for stage 2 and this is for stage 1. I am not going into details, but it is on the slide. It's when you are done with your calculation by looking into how many CO2 has generated, how many O2 will be used, how many ATP will be generated. This is how it will look. But it is all following this basic stoichiometry that we have just covered. Okay. Now, I will not go into much detail, but I will quickly show you that there are different other fatty acids that they have their own trajectory. I spoke to you about MUFAS. I spoke to you about PUFAS, mono. What also happens is odd carbon and even carbon. We have discussed about palmitate, 16 carbon, myristoyl or myristate, which is again 14 carbon. What about the 13th carbon? What about the 15 carbon? They can also have their own trajectory. On your slide right now is mono unsaturated fatty acids. So if you look into the mono unsaturated fatty acids, okay, which is basically one double bond. So this is oleoyl coenzyme A. So what you see here is, first of all, it will have, so the double bond is here and the coenzyme A is here. Okay. So you have 3 cycles, one. So in the first cycle, this go, in the second cycle, this go, in the third cycle, this go. In the fourth cycle, you have this double bond which needs to be broken. Here, there was no double bond. So there was no double bond. Initially, there was no double bond between the carbons. So there was no enzyme that can deal with that can break the carbon bonds here, right? There is no need. But here, you need this removal here. So first of all, till it reaches the double bond, there will be beta oxidation in the normal way because they're all single bond, and you will have 3 acetyl coenzyme, 1, 2, 3. Okay. Now, what happens is it has encountered this double bond. So this double bond will be encountered by an enzyme called cis-acting delta 3 dodecanoyl coenzyme A. So this is your structure. So this will be acted upon by delta 3 delta 2 enoyl coenzyme A isomerase. What it does, simply, it is from the cis configuration, it brings it to the trans configuration. And why is it important to do this? Because when it is in trans configuration, here, this enzyme can start acting. For this enzyme, it is in trans configuration. So see, this is cis config. This is both the hydrogen on the same side. So when it is in this form, it just bypasses the first step. It doesn't need to do this. It is already formed a double bond. It needs something which makes it into a trans configuration. So it uses isomerase. This is in trans configuration. And once it is in trans configuration, it can simply continue with this step. So here, after this, you require 5 more cycles to give 6 more acetyl coenzyme, right? So what is the idea here? If you have a double bond with their in cis configuration, you just till the breakdown is happening, till reaching this double bond, you have your normal beta oxidation. Then you have the incorporation of an enzyme which makes it from cis to trans configuration. Once it becomes trans, then the normal enzymes which are of the fatty oxidation pathway, we can act and it can give you acetyl coenzyme. Right? So this is mono. But what happens when there are poly? What is poly? Poly is basically multiple this was just one unsaturated bond. So you can add this enzyme and this can work. But what happens when you have multiple? Now, what happens when you have multiple is, so let's say again, this is fatty acid. Now it has here as well as here. So first of all, again, 3 of these are removed. So you come to this structure now. Okay. Now, when you have this structure, you have again these isomerase that we have just discussed. Now, when you have this isomerase, this makes it into trans delta 2 and cis delta 3. So you have one beta oxidation cycle and first oxidation of the second cycle. So you have oxidation acetyl coenzyme A of this hydrogen of this bond here. Then you have this structure. Now you read one additional enzyme which is reductase to reduce it, to bring it to one single bond. to reduce it to a double bond from this to this. And then you have this isomerase, which mixes in trans configuration, and then you have acetyl coenzyme A synthesis. Don't get confused. It's just that in the first step, you have isomerase, but because there are two back-to-back unsaturation, where there is back-to-back two double bond, you also need a reductase, and then you again use this isomerase. Okay. Yes, a bit complicated, but I am sure you're understanding it. So when you are revising it, these are the things that you need to keep in mind. Okay. And this is what I was saying. If you have odd numbered fatty acids, let's say propionyl, so you basically, what you do is, in this, the coenzyme A, fatty acid coenzyme A, they are acted upon by carboxylase enzymes. So the carboxylase, which bicarbonate and biotin, make them into something called methyl malonyl coenzyme A. And this methyl malonyl coenzyme A, when they are acted upon by epimerase, they give you from D to L, and from L, in the L configuration, when they're acted upon by methyl malonyl coenzyme mutase, they give succinyl coenzyme, and we know where succinyl coenzyme is utilized. So when you have an odd numbered fatty acid, the end product is succinyl coenzyme A, that is used in, as we know, citric acids, as we know, that is used in the citric acid cycle, right? So this also gives you a, how do you say, intermediate, but not acetyl coenzyme A, but succinyl coenzyme A. So odd numbered fatty acids have a different target, fate, but the others make use of these basic four enzymes. But in case of mono unsaturated and polyunsaturated, there are additional steps. In mono unsaturated, you can remember it, mono unsaturated means one carbon, so one additional step, which is isomerase. In polyunsaturated, because it's poly, so you have three additional steps. First the isomerase, then the reductase, and then the isomerase again. Okay. All right. And lastly, as we have already discussed multiple times, when there is fatty acid oxidation or any regulation is there, there is a crosstalk between the anabolism and the catabolism. So on the right, you have oxidation, which is basically breakdown. On the left, you have the synthesis, which is basically making. So you have to have a crosstalk between these to regulate the amount of fatty acid that you can see. There is this glucagon, as we have already discussed. So glucagon here is inactivating the fatty acid. So if you, when you are reading these figures, just be mindful of the underlying principle here, that the idea is your cell requires energy. If your cell has no energy, catabolic methods or the breakdown pathways will be in activation. If your cell has energy, you don't need any more fatty acids, so catabolic methods will be stopped, or the pathways which are involved in breakdown will be stopped. Hello, am I audible? Sorry about that. Yeah, sorry about my network. Okay. Okay, so we have discussed a lot about fatty acid oxidation, and what I was saying is basically you just need to remember the idea in the mind that if you need energy, you have breakdown of the biomolecules. If you don't need, if you are excess on energy, then you have storage, right? So we have 5 questions before we move on to the second part of today's session, and we'll do this quickly so that we can move on to the next topic. Okay. Yeah, we have, I can give 10 minutes to you for these questions. So maybe you can read these questions and we can discuss the answer after this. Okay. So the first one, just a second. Yeah. Okay. So the question is, I will come back again. So question says, an in silico study identified a drug which is called drug X, and this drug X, as we know, has potential to bind and phosphorylate certain proteins. Now, the phosphorylated form of these proteins stimulated lipid breakdown. Fantastic. We know that this drug phosphorylates the protein which are involved in lipid breakdown. And we are just supposed to know which ones are getting phosphorylated for lipid breakdown among this. And this is the first thing that we have discussed. Remember, when we had the hormone, we had adenylyl cyclase, we had CAMP. CAMP went to something called CAMP dependent protein kinase A. Protein kinase A means something that adds phosphorylate group. And there were two things that it was phosphorylating: carbon sensitive lipase in the cytosol and perilipin, which is on the lipid droplet. So A and B, not protein kinase A. Protein kinase A is not getting phosphorylated by CAMP. CAMP. So the protein kinase A is dependent on the CAMP. If CAMP is there, it binds to CAMP and it gets activated. It basically has this function where it can phosphorylate the downstream components. Okay. Okay. Next one. So we have already discussed about many things about fatty acids. So the question here is, which of these are false? B and D. Okay. I will show the answers and then we can look into. So we are discussing about the false statement. So B and D are correct. We also have C as the false statement, right? Let's first look into this. So it says that saturated fatty acids undergo beta oxidation. Of course, yes, because we have just discussed about this, right? Okay. Second one says the fatty acids are broken down in the mitochondria one carbon at a time. No, two carbon at a time. Remember acetyl coenzyme A, and then the remaining backbone. So two carbons, acetyl coenzyme A. Right. The third one says omega-3 fatty acid means a double bond exists between the second and the third carbon, which starts from the methyl group. So when you look into it, you will see that in omega-3 fatty acid, which is like this, the first double bond uh, is between the third and the fourth carbon from the methyl end. Okay, and so the structurally it is in the third and the fourth carbon, but from the methyl group. So that's why the second, the next one is also incorrect, because it says there is a double bond which is between the third and the fourth carbon, but it is from the carbonyl group. No, it's from the methyl group. So when you see it from the other end, here you will see that it has the omega-3 fatty acid. And lastly, only fatty acyl contains containing 12 carbons require shuttle, yes, because they are bigger in size. Okay, next one. So in this, we are talking about basically the different steps that we have just discussed about beta oxidation. A and C. Okay. So we're talking about correct statements. Okay. And the first one is the only incorrect statements. Okay. We'll see why. The first one says conversion of palmitoyl coenzyme A to trans delta square enoyl coenzyme A requires hydratase. So if I go back here, you remember this is what they are talking about. So the enzyme here is dehydrogenase. Okay. Okay, let me go over it and then I'll come back to it because I'm since here on the slide. The second statement is conversion of L beta hydroxy acyl coenzyme A to beta keto acyl coenzyme A requires beta hydroxy acyl coenzyme A dehydrogenase. Right? So dehydrogenase, hydratase, and dehydrogenase. So this is correct. Third said in beta oxidation of palmitoyl coenzyme A, NAD plus is converted to NADH and H plus. So this C statement is also correct. B is correct, C is correct. Now, D says the final product of beta oxidation of odd number fatty acid is fatty propionyl coenzyme A and acetyl coenzyme A. This is where we were. So this propionyl coenzyme A is an artifact of the oxidation of odd number fatty acids. So the even part, or till you encounter the last carbon, you will have acetyl coenzyme A, and the last one will give you propionyl coenzyme A, which will give rise to succinyl coenzyme A. So you have acetyl coenzyme A, and the propionyl gives you succinyl coenzyme A, right? So this is why this has a D. So that's why, remember when I was discussing in the previous session, I said I can skip some topics because those topics will crop up in your questions, and in with solving them in questions make us more easy to understand. So with one question, it became easy that in the odd numbered fatty acid, you have first you have all the acetyl coenzyme A, and the last ones which are remaining, they form the propionyl. Propine means three, the propionyl coenzyme A that gives rise to succinyl coenzyme E. So basically, fatty acid overall odd number fatty acids gives you acetyl coenzyme and succinyl coenzyme. Okay, next one. More you solve questions, especially such MCQ based questions, more these topics will come in your grasp. So it basically says even numbered carbon. So yeah. C and G. Okay. C, yes, G, S. Enol enzyme isomerase as well, right? So there they remember they are used in the unsaturated even number fatty acids. Okay, so I will go back here. Just a minute. Yes, here, right? So when I said even numbered, I had incorporated both saturated and unsaturated, both of the types. So that's why Enoyl coenzyme A isomerase, and these we already know are involved. Methyl malonyl, all are involved in your um, odd numbered fatty acids. And this one here, 2,4-dienoyl coenzyme A reductase, it's used in polyunsaturated fatty acid, but when it's present in the double bond is present in both even and odd positions. So that's a bit more complicated, not only even numbered. Okay. All right, this one. Which of the following enzymes cofactors function in one or more fatty acid oxidation pathways? So basically, which are involved in fatty oxidation pathways. B only. A, B, C, okay. That's because, okay, let me go back. I think you will see it apparently. NAD, so I think we have already discussed. Biotin and coenzyme B12, both, all of these are here. So biotin you already told. So vitamin B12 is also used here, which is basically the cyanocobalamin. So, sorry. So this three I used. NAD, we've already done this, right? We've discussed this. It is part of the pathways. Acetyl coenzyme is product, and TPP is not really used in fatty acids. We have discussed TPP the previous session, but not today. Last question before we move on to the next part for today's session. It's about the regulation of fatty acids. So think about the, like the products, and then think about will it activate or will it inhibit? Okay. B and E. Okay. Exactly. Great job. Right? So if we look into the first one, which is malonyl coenzyme A, and it says that it stimulates the carnitine-mediated transport of fatty acyl chains, it is basically incorrect because malonyl coenzyme A inhibits carnitine transferase. So carnitine is what, so as we know, carnitine mediates the transport, and the enzyme there is carnitine transferase, and malonyl coenzyme A inhibits this. So it doesn't stimulate the transport. Second is correct that we have already discussed. Acetyl coenzyme A carboxylase is regulated by phosphorylation, dephosphorylation. In case you have missed it, it's here. So it's phosphorylation by pKa, which activates it and inactivates it. Malonyl coenzyme A, it inhibits the transferases. And then it said that third one said low blood glucose level, which is here, stimulates acetyl coenzyme A carboxylase activity. If you see this, it rather makes it inactive. Okay, so low blood glucose does not stimulate, it inhibits the enzyme here, right? Fourth one, coming back to the question again, fourth one said that fatty acyl chains in the mitochondria are transported in the cytosol to synthesize membrane lipids. Well, that's not correct. For membrane lipids, citrate is what is exported from mitochondria. So when you see the synthesis of membrane lipids, you will see that citrate is what is exported out. And lastly, this we have already discussed. Phosphorylation of perilipin is what enables the lipase to come and act on it to release the lipids. Correct. So let's quickly move to the next part, which was biosynthesis. Since I already told last time also, if we are breaking it, we should also make it. So talking about fatty acid biosynthesis, which is basically uh, synthesizing the fatty acids, synthesizing the fatty acids in a stored form, right? So when you look into the biosynthesis of fatty acids, unlike glycolysis or unlike amino acids or unlike any other biomolecules, these two steps don't go in the reverse manner. Basically, the components that we just read in fatty acid oxidations are not the same in the reverse direction in fatty acid biosynthesis. Fatty acid biosynthesis has its own trajectory. And one of the main component which is used in fatty acid biosynthesis is malonyl coenzyme A. This malonyl coenzyme A is formed from acetyl coenzyme and bicarbonate. And the enzyme which makes it is acetyl coenzyme A carboxylase, as you can see on the slide. Right? So this acetyl coenzyme A carboxylase that you see on the slide, it has, if you see in the central three colored part, if you see, this has three regions. One is this biotin carrier protein, which is in gray. Then you have the biotin carboxylase, which is in the blue, upper blue. And this step here, this part here, catalyzes this action here. So it utilizes the ATP, and your bicarbonate is incorporated into the backbone of biotin. Okay. Then, the next part is transcarboxylase, where you have the acetyl coenzyme A added to this backbone of malonyl coenzyme, to the backbone, which gives you the malonyl coenzyme A. So you have first incorporation of bicarbonate, second the incorporation of acetyl coenzyme. Together they give you malonyl coenzyme A. And this is very important for lipid biosynthesis, right? And we'll see how. Okay, so when you see lipid biosynthesis, there are 2-3 things that will come very obvious to you. One thing is that all this biosynthesis, when you see, they will take place in this structure here, and they will be again like fatty acid oxidation. They will be step of 4 series of 4 steps that will be repeated in continuous manners. Okay, so we have the malonyl coenzyme A, and it's important because that is how it will begin the entire process of your fatty acid chain synthesis. Okay, so this slide on your or this slide here shows the steps which are involved in addition of growing or growing fatty acid chain, which will be added, which will be incorporating the addition of 2 carbons. Okay, so we'll see how. Let's look first here. You have malonyl coenzyme A, which is the very first thing that got incorporated, and you have the acetyl coenzyme A. If I go back to malonyl coenzyme A, this is the structure. Remember the color codes here. This is in green, the carboxyl group. This is the acetyl coenzyme A backbone. Okay, together they make up malonyl coenzyme A. So you have malonyl coenzyme here, so malonyl group and the acetyl group. Okay, now the enzyme that is used or that is catalyzing this entire synthesis is called fatty acid synthase, and we will look into this circular structure in a while. But what you now need to remember for now is there are these 2 chains that is where these growing fatty acid chains are incorporated, right? One is here and one is here. What are these 2 places? So we have one. So these are both thiol groups, okay? And they are, so these intermediates that we will see in a while, they remain bound to these thiol groups here as thioesters. The first attachment point is again a thiol. As we know, thiol group is SH. So SH group of cysteine residue, because cysteine is S. So that SH is in the one of the enzymes component or one of the protein, which is called beta ketoacyl ACP synthase. That is smart for you to remember the name right now, but just remember that there is one of this, which is basically this, is one of the proteins of the synthetase. The other is a acyl carrier protein. So these both are component of fatty acid synthase. They both have these thiol groups, and both the thiol groups are where these chains are forming. Okay. So one of them have attachment of malonyl group. The next one has acetyl coenzyme or acetyl group. What is the first thing that takes place between these two in mixed proximity? There is condensation, and you have this green group removal. So what happens is this group attacks this acetyl coenzyme A, and this gets transferred to this chain. So this entire thing moves to this acyl carrier protein. Okay. So this is the first thing that takes place. Your acetyl coenzyme or acetyl group is attached with the other group of malonyl group. Okay. Now this is your first step. After this, you have reduction. Reduction means you are oxidizing NADPH using that H plus, and you are basically reducing this carbon here, carbon oxygen bond here. So you are making CHOH. Third is dehydration. Dehydration means you are now removing water. So you can remove this water, so OH from here and one H from here. It makes it a double bond, CH in trans configuration. Remember, trans configuration is very important. So trans configuration. And lastly, again, you have a reduction. Again, you basically reduce this, and the use of the NADPH, and you remove this double bond as CH to CH2. So you have a saturated acyl group which has 4 carbon. Okay, this is the first step. And this, if this is clear to you, then the remaining additional steps becomes very easy to understand. So just remember it once again. 4 steps: condensation reduction, dehydration reduction. Okay. First step, you have malonyl coenzyme and acetyl coenzyme A. They both are getting in a growing chain. So you will also see these kind of growing chains if you have studied about translation, where your growing peptide is forming on one end and then it is transferred onto the other chain which is already sitting at the other side. If you remember translation, similar kind of machinery is taking place. You have this growing chain here. So you have the new incoming group here. All these groups are attached and transferred to this other thioester here, thioester linkage group here. But while this transferring is taking place, there are 4 steps that take place: condensation, reduction, dehydration, and reduction, to finally give you 4 carbon compound, a 4 carbon chain. Now in the next steps, we'll always have this 2 carbon which is being added to the chain, because the 2 carbon will come from acetyl group. So again, if you see this here, again here you will have, so let's, where is the second round? Where did the second round go? Yeah. So we'll see in a while how the second round takes place, and these will keep increasing. Okay. All right. But you have understood the first round, right? So if you look into this overall, so this is the 16 carbon compound. Now remember one very important fact: when there is fatty acid synthesis, only 16 carbon, till 16 carbon fatty acid can be formed. Only till palmitate you can have the synthesis. There is no synthesis of fatty acid beyond 16 carbon, and we don't know why. But you only have the synthesis of 16 carbon member. Okay, among which the first 4 steps of we have already established. Okay, we have already seen it. Now, if you take the overall process of palmitate synthesis based on what we have just seen, so basically you need 16 carbon. So this is the very first step, and this here is the chain after the first incorporation, right? First incorporation of acetyl group and malonyl group. So this is the first chain. Now see, if you go back here, it's very evident to you. This was where we stopped, and this in the second round, it is here. It's here. So this transfer is also taking place from this group. This is getting transferred to this group here. So why will that happen? We'll see in a while. But remember, the first chain is made here and it is transferred to this other thioester. Okay, other thiol group. Now this thiol group is again free. So you have another, which is, what is this enzyme called? What is this thing? This is a malonyl coenzyme. So you have a malonyl coenzyme. Again, the CO2 is removed. This 2 carbon is added to the growing chain. Okay, so they are added here below. Again, they are added, they're taken here. So this way you are adding till you have 16 carbon, which is you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Now, of this 16 or the 14 or the 12 carbon fatty acid, the first 4 will be from the first step of the synthesis, and the remaining 2 carbons will always be from the subsequent round of synthesis. Okay, you can see from the color coding here. We had acetyl coenzyme A that begins the group. We have malonyl coenzyme A, that is the next part, and then we have all these incoming CH2, 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 which will be added to the growing chain. Okay, all right. Now, before I go to the second round, let's make it a bit more complicated for you, but it's not complicated. You just need to see the structure of the, I told you the circular structure, right? So if you just overall, if you look into the structure, you have this SH groups here. This is what I told. One SH is from the carrier protein, acyl carrier protein, and the second is from these enzymes. So all these are different enzymes. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So there are 6 proteins, 6 such segments which are part of the overall enzyme called fatty acid synthesis or fatty acid synthase. Okay, so at each point, as you can see, so what you need to quickly look into is this blue colored. So what it means is wherever you see blue colored, that enzyme or that enzyme basically is what is active in that step. So first is this, then is this, then is this, then is this, then is this. Now, this is the overall figure. I don't want to explain from here because it makes it more complicated to understand. But we've already looked into the 4 steps together, right? So we'll quickly take a look into these steps individually. Right? We know the first step is condensation, which is removal of CO2. So this is the first step here, if you see. But what about these steps here? Remember, we have discussed that you need acetyl group and malonyl group. This is basically acetyl group transferase, malonyl group transferase. Acetyl group being transferred here. This empty gets activated. Malonyl group is transferred here. These two enzymes, these two are coming together. So before your first step, you have 2 enzymes which have transferred the required group onto the 2 locations. Now the first thing is condensation, which is basically removal of CO2. So from these, we have already seen CO2 is removed. The enzyme that is involved in this is something called beta ketoacyl ACP synthase, which is KS. See, the KS is the location where you have the acetyl enzyme coenzyme A group added, and this is the first enzyme that takes place in the stage 1. So that is how you can remember also, right? So what takes place here is the acetyl group is transferred from the cysteine SH to the enzyme malonyl, and this becomes the methyl terminal 2 carbon, which we have already discussed. So not so complicated. So now you have your condensation takes place. Now you have your product ready for the next step. The product is called keta-butyryl ACP, and the enzyme which is now getting activated is KR. KR, if you see the name, it's ketoacyl ACP reductase. The product is ACP and the enzyme is reductase. And we know the step which is involved here is reduction. So a reductase enzyme is getting into action and it is reducing the carbonyl group which we have already established. And from there it is give you, it is the product which is coming up is beta hydroxybutyryl ACP. I am not explaining the names. I'm not explaining everything in detail because that will take a long time. We don't want that. I'm just telling you in a brief overflow. When you are studying, it will be very logical for you to follow this. Okay. So you have a reduction and you have, but I want you to understand from these figures when you go back is you need to know how to read it. So you have KR activated. Now, after the second step, this is the product. And now the HD enzyme will start activating. And we know the third step is dehydration. So this is dehydration. HD is hydroxyacyl ACP dehydration. So it is now getting dehydrated, forming something called trans delta square butanol ACP. The enzyme now getting activated is ER, which is basically again a reductase. So it is getting reductase. So the same chain of reaction, condensation, reduction, dehydration, reduction is what is taking place with their enzymes in action. Okay, so when you have this reduction, you have butyryl ACP, which is the first 4 carbon which is added to the growing chain. Then this chain is transferred to this group here, and then you have additional carbon coming in. Okay, right. So this is what is happening. So you have the translocation of this butyryl to this cysteine on the beta. So from this, it moves to the Ks, and this remains free, where you have the incoming malonyl coenzyme A. This is what you need to remember very, very briefly. 2 points of attachment. Acetyl coenzyme A, malonyl coenzyme A, they forming the first 4 carbon on the acyl carrier protein, uh, SH group. Then it is transferred to the other part, and this new malonyl coenzyme, the remaining, the subsequent addition or formation of fatty acid is by malonyl coenzyme. It looks very complicated. It isn't. I'm just giving you a heads up, right? So again, you have second round. You have the malonyl coenzyme who will take part, malonyl transferase. It will transfer. Now, what will get bound to what? Your carbon dioxide is removed. This group, remember so when the first stage is taking place, when the first step was taking place, or maybe this is clear here. So when the first step is taking place, this basically is moved to here. And after the completion, this moves here. So there is a circular movement here. This is what is happening here also. The first step, this gets attached here. I mean, after the second round incorporation of malonyl coenzyme, this thing gets transferred here. And after they are fully formed as a chain, this gets transferred to the Ks. You need to just remember in a flow, it will become easier for you to understand. Okay. And this is the stoichiometry. Basically, it tells you how much, again, acetyl coenzyme A was used. So you have 7 acetyl coenzyme A being used. And each acetyl coenzyme A, whenever it is incorporated, you have a CO2 getting out. And you have ATP. What is the ATP being used up? We didn't see that. So the ATP is basically used up when you have this steps here. Okay, and when you look into the internal use, so you also have an ATP usage. So you have ATP which is being formed to ADP and PI, right? Now, this is the formation of several malonyl coenzyme molecules. This is the first. Oh, I'm sorry. This is the first step here. This is the first step here. Now, once you have malonyl enzyme A and coenzyme A, this together will give you palmitate. So you have 1 acetyl and 7 malonyl coming together to give you palmitate. And ultimately, if you join all of this is the stoichiometry. Again, I'm not going into details. I'm pretty sure you will be able to understand it, but it is on the slide. You can check it whenever you feel like. And again, coming back to always, your regulation is again determined by the products. So if you have malonyl coenzyme A and palmitoyl coenzyme A, palmitoyl coenzyme A stops this reaction here from this getting converted to malonyl, because when malonyl coenzyme A is produced, it is basically lipid biosynthesis will take place. But when you have lipid already, you don't need this conversion to take place. Similarly, you have glucagon, epinephrine, which are basically saying there's low blood glucose level. So if there is low blood glucose, you need energy. So this synthesis pathway is stopped. It rather activates these pathways. Okay. All right. Now we have 30 minutes, which is fantastic because we have a lot of questions, and I will go easy. We'll not rush it up. So basically what I have is 5 questions from this topic, and then there is a set of 10 questions mixed and matched from both, which will help you grasp more on this topic. And so I would say we'll try to do as many questions as possible till our time permits. And then I will, if time is remaining and question is also remaining, we'll just quickly look into the questions, right? But let's go easy on this and let's not try to jump into it, right? And as Aditi has already mentioned, the answer here is A is what she says, but let's look into this, what's exactly happening here. So the question here is, which of the following statements are true regarding biosynthesis of fatty acid? So the source of carbon dioxide or biotin carboxylation is bicarbonate that we have already established. Carboxylation of biotin is ATP. This is just what we have seen here, right? Okay. Next one says carboxylation of biotin is catalyzed by acetyl coenzyme A carboxylase, which is also correct because that's the enzyme. If you go back here, this is the enzyme. And then the last one says transcarboxylase is responsible for the formation of malonyl coenzyme A. So when you go into the first slide, this one here, this is what is actually forming the malonyl coenzyme A. So among that enzyme also, transcarboxylase domain is what is responsible for this malonyl coenzyme A. Last one says the formation of malonyl coenzyme A is reversible. That is incorrect. It's not a reversible. It is an irreversible, highly regulated step of fatty acid synthesis. Okay, next one. Now, next it says about the incorrect options out of these. The question says, but which of these are incorrect? The first one says CO2 is released during condensation of acyl coenzyme and malonyl coenzyme A. Next is a product of condensation reaction is a 5-carbon acyl chain. Yeah. Next one says CO2 is released from the 5-carbon product to yield a 4-carbon acyl chain. Next it says fatty acyl chains are extended by 2 carbon units in each cycle. And the last one says that the addition of carbons to the chain is a 4-step process. B and C. Okay, just a second. Exactly correct. So the incorrect statements are B and C. So first one says CO2 is released during the condensation of acyl coenzyme A and malonyl coenzyme A. We already have established there's a condensation. That's the first step of fatty acid biosynthesis. Second one says the product of condensation is a 5-carbon carbon acyl chain, which is not correct. It's a 4-carbon. That's how we have established, right? We have 4-carbon acyl chain at the first one. Third says carbon dioxide is released from the 5-carbon product to yield a 4-carbon acyl chain. That doesn't happen. There's no 5-carbon acyl chain. D says it is extended by 2-carbon, which is what we have already seen. And the fifth one says it's a 4-step process. That's correct because that's how we have condensation, reduction, dehydration, reduction. All right, great, fantastic. Next one. Similar question, which of these takes part in fatty acid biosynthesis? So you have this enzyme, malonyl coenzyme ACP transferase. All these names are part of the fatty acyl synthesis that we have just looked into. What you need to do is you need to identify which ones actually are part of the enzyme complex. Okay, maybe I can give the answer. Okay. Wait. Sorry. Yes, so the first one is correct, malonyl coenzyme A ACP transferase. Second is correct, beta ketoacyl ACP transferase, and enoyl ACP reductase is also correct. The fourth one is also actually correct. I don't know why I didn't pick it, but the fourth one here is also correct. Maybe I'll just show it here. We know this is the first one. Okay, butyryl is incorrect, and trans 2-butynyl ACP dehydratase is also correct. So maybe I'll just go back to this step here. So the name of the enzymes are actually in the first one that we have looked into. So we have the first enzyme is malonyl coenzyme A ACP transferase, that is here. Wait, sorry. Okay. The second option was beta ketoacyl ACP reductase, which is here. We had enoyl ACP reductase. We had beta ketoacyl ACP synthase, which is here. Then we had beta ketobutyryl ACP reductase, which is incorrect because we have hydroxyacyl reductase. And then we have trans 2-butynyl ACP dehydratase, which is again not part here, which is basically you do have dehydratase, but that's a hydroxyacyl. Okay. All right. So that question was yeah, CAB, right? CAB, but you also have ketoacyl synthase, right? You have it here. ACP synthase. So what I think is important here is that since this doesn't have ACP, that's why it's an incorrect name. But if you look into the what do you say, overall name, it's correct. But since it doesn't have the absolute name of ACP, that's why I did not point it out as an correct statement. Okay, next one. Which statement about this is incorrect, which is basically the acyl carrier protein, which is part of the, which is where the SH group is for the synthesis of fatty acids. It says that it is a vitamin B5 derived coenzyme. It has an alcohol group where the acyl chains bind. This is very giveaway already. Next one says it catalyzes the conversion of malonyl group to hydride. And the fourth one says the incoming malonyl group binds the sulfur hydride group. This one's easy. So yeah, we'll do it quickly. B and C. Says incorrect. Absolutely correct. Fantastic. The first one says it has a vitamin B5 derived coenzyme, and the coenzyme or that prosthetic group is called 4' phosphopantothene, and this comes from the pantothenic acid, which is P5. So it is correct. Next one says it has an alcohol group where the acyl group binds. B and D are contracted to each other. And D is correct because we have already just established that it binds via the SH group. So the incoming malonyl group binds by a sulfur hydryl group and not by the alcohol group, right? And third one says it catalyzes the conversion of malonyl to hydride. Well, it doesn't catalyze anything because this is a carrier protein. It just carries the group. It doesn't catalyze any reaction. Okay, next one. Next one says, again, based on this, which statement, which one of these are correct? So it says acetyl coenzyme A exits the mitochondria as malate. The conversion of malate to pyruvate by malic enzyme requires NAD plus reduction to NADPH. Citrate positively regulates the activity of acetyl coenzyme A carboxylase. Palmitoyl coenzyme A inhibits acetyl coenzyme A carboxylase. Insulin positively regulates citrate lyase activity. And acetyl coenzyme A carboxylase is inactivated by phosphorylation. Out of this, which statements are correct? Uh, is the F only for this option, or do you only F? Is that your only answer? What is the question says correct? So you say F is the only correct statement. If you have trying, then should I wait? Then maybe I'll wait for 1 minute and then we can discuss the answers. Think. There is a lot of crosstalk here, right? So just think, will this be important? Will it be activate? So out of this, let me tell you, there are at least two that we have already discussed in the regulation. Okay, no problem. Don't worry. Okay, so this one is, these are the correct, basically all of these are correct answers, right? Except for the first one. So let's first understand the first one. The first one is acetyl coenzyme A exits the mitochondria at malate. So when you see into how acetyl coenzyme A is transported, you will see that acetyl coenzyme A again cannot cross mitochondria on its own, right? So it does not move as malate. What it rather does it acetyl coenzyme A and oxaloacetate from citrate, and that citrate is what is moved out of the mitochondria. And once in the cytosol, you have this enzyme which is where did it go? Citrate lyase, and this citrate lyase basically cleaves the citrate, and it basically gives the acetyl coenzyme A back. So the cycle is acetyl coenzyme A and oxaloacetate come together to form citrate, and then this moves out of the mitochondria to cytosol. In the cytosol, you have this enzyme citrate lyase, which basically cleaves the citrate again and gives you acetyl coenzyme A back. So that's a cycle of it. So it's not malate, but it's rather as citrate. Okay, and with that, this statement is again correct. Insulin positively regulates this activity. Okay, so when I say positive, I mean if citrate is if insulin is there, citrate lyase activity will increase. Okay, when I say negatively regulated, it means if I have more of insulin, then my citrate lyase activity will be less. So inhibition. Okay, negatively affecting is inhibition. Okay, all right. Now the next one says B, conversion of malate to pyruvate or pyruvate by malic enzyme requires NAD reduction to NADPH, which is correct. Next is citrate positively regulates the activity of acetyl carboxylase. So when you go back in this slide here, you will see this is correct. This is positively regulating it, right? And then we have palmitoyl coenzyme A inhibits acetyl coenzyme A carboxylase, which is also something that we had already looked into, right? Then it says insulin we have already discussed. It says acetyl coenzyme A carboxylase is inactivated by phosphorylation. If you remember the very last slide of here, this one. So if it is phosphorylated, it is inactive. So it is correct. Yeah. Okay. Any problem? Did the answers make sense? Okay. All right. Next. Now I think we are in this hybrid mode where we have a lot of questions of synthesis and oxidation, which is a good exercise for us to go over these topics together, right? Okay. We'll take it slow. We have some 20 minutes, so we can take it easy. Okay. Now, which other step is the first step in the oxidation step of saturated fatty acid? Okay, thank you. D, correct. See, it's easy, I told you. Next, which of the following are true about oxidation of odd chain fatty acids? So you have odd chain. Remember, we discussed about this one intermediate that gives rise to succinyl coenzyme A, and the others give rise to acetyl coenzyme A. So I did not discuss it in this case because we had a question where we can discuss about this. ABD, ABD. Okay. C also. I will show you the slide once again. So A is correct, produces propionyl coenzyme A, which finally goes to succinyl coenzyme A. C is also correct. We have this enzyme. I will show you in a while. And D says it requires vitamin B12 and biotin. And E, TPP does not take part in fatty acid biosynthesis or oxidation. We have already established this. Let me quickly show you the propionyl coenzyme slide. We have this enzyme here. Okay, and we have the biotin here. So this mutate is also there and biotin is also part of it. Okay, yeah, but you did it all right. So wait a minute. Yeah, okay. Next one. Which enzyme catalyzes the final step of beta oxidation? It's a good place for you to try to remember the entire pathway and see which one, what's happening there. Try to remember in oxidation what's happening. How did we start with? What you need to do down to give you acetyl coenzyme? What was happening in? Okay, C. Correct, absolutely correct. So the last step is by ketothiolase, which what is it doing is basically cleaving the beta ketoacyl coenzyme A, which gives you the acetyl coenzyme A and the other acyl coenzyme A, right? So once again, if you quickly remember the steps of the beta oxidation, you had the dehydrogenation, where you have the FAD-Ling oxidation. Then you had the hydration step where you had the water incorporation. Then again, you had the dehydrogenation where again there was NAD linked oxidation. So the first was FAD linked oxidation. Third was NAD oxidation. And the last step is this thiolysis group where you have the acetyl coenzyme as one product and the other acyl coenzyme as the other product. Fantastic. Okay, next one. Read it easily. It says in a fasting state, which of the following would you expect to occur in adipose and liver tissue? Basically, the fats. If you are in a fasting state. So try to understand if you are in a fasting state, what is the body demanding? What are the cells demanding? You can remember it as, do you will the cells like more or will the cell uh, push towards catabolism or push towards anabolism, synthesis or breakdown? Yeah, B, C, D. Okay, exactly that. Fantastic. Absolutely correct. So the first one says acetyl coenzyme carboxylase activity will be increased. This is incorrect, because if that is increased, then you have more of like the synthesis pathway taking place. So your acetyl coenzyme carboxylase will be actually inhibited, which will reduce the malonyl coenzyme A. And if you have less of malonyl coenzyme A, you have less synthesis, right? And the second one is correct, because you have phosphorylation of perilipin, then you have mobilization of the stored fat. Third is again correct, because it is synthesis is inhibited. Fourth is transport of fatty acid chains into mitochondria, where your oxidations will take place. That's correct. Fifth is again incorrect, because it says there is increased TAG synthesis. Again, there is no synthesis here, right? So what actually happens is, when you are fasting, you have low insulin, high glucagon, and these enzyme would rather have more of energy sources in the cell. So they would activate, or they would facilitate all the enzymes that are involved in the breakdown of the lipids, right? So great. Okay, next one. So it says about something called peroxisomal fatty acid oxidation. This is again something we haven't discussed. So I just wanted to keep in it here, so that you get a overview. So you can try attempting, and then we will of course look into the answers. So we are looking here into peroxisomal fatty acid oxidation. You can try to attempt, and of course we will be discussing, so that won't be an issue. So peroxisome, if you read, or if you study, you will see one word which is very concomitant with peroxisome, which is peroxide, right? So that should give you that should help you understand some of the options here. Okay, BDSC. All right. Okay, BCE. Okay, and we will see. We are looking into the correct statements, so we will see which ones are correct. Like, which is C. Okay, you said C. That's correct. That's exactly what it is. But B and E also. So let's first try to understand peroxisome fatty acid oxidation. Okay. So when do fatty oxidation takes place in peroxisome? Because we know it takes place in mitochondria, but peroxisome, in peroxisome, it basically the very, very long chain fatty acids, which are greater than 22 carbon. We have discussed about 16. We know about less than 12 being easy to move inside. When we have very long fatty acid chain, which is 22 carbon, that's when your peroxisome come into action. Okay. And that's why they are they are oxidized in peroxisome to a medium chain fatty acid, such that that fatty acid chain can now be used by mitochondria. So peroxisome basically reduces the length, and it goes further to mitochondria, where further steps take place. Now, while it is being oxidized in the peroxisome, you have peroxide, which is a byproduct, which is hydrogen peroxide. And this hydrogen peroxide, if you know, it's very it's it's problematic for the cell. It can give rise to free radical. So that peroxide is tackled by something called catalase enzyme, which basically breaks this peroxide into hydrogen and oxygen. So it breaks it up, right? So what you need to understand is peroxisome fatty acid oxidation takes place for a very large fatty acid groups, like more than 22 carbons, and produces hydrogen peroxides, right? But it is not the alternate for mitochondria. It basically just reduces the length of 22 carbons, say, to a medium length 16 or 18 or 14, and then it passes on to mitochondria where the actual oxidation takes place, right? So that's why B, C, E are correct. First one is incorrect because the name suggests it's peroxisome, so it's not mitochondria. Mitochondria and peroxisome are two different organelles. And unlike mitochondria, here you don't need the carnitine shuttle to move the fatty acid coenzyme inside. Okay, clear? Was that okay? Yeah. While you, so the next questions are set of question is, which of the following are true about fatty acid synthesis in humans? Okay, this one's also easy. We have already discussed most of it. So you basically, it doesn't, so basically what we have discussed so far is what is going to be the workflow or logic that you should be thinking. And you already know all of these. DEA. A, yes. D, yes. Not E. I will come to it. Wait. So the first thing is correct. It happens in cytosol. We know this already. Synthesis, right? Second is NADPH is a reducing acid agent. We have seen this. D says it uses malonyl coenzyme A as a 2-carbon. We have already seen this, right? Third one says acetyl coenzyme A is directly transported from mitochondria to cytosol. We have established, we have already discussed this in one of the previous questions, that for acetyl A to be transported, it has to be transported as citrate. So it combines with oxaloacetate to form your citrate, moves out from mitochondria to cytosol, and there it is converted by citrate lyase to give you the acetyl coenzyme A. Okay. The last one says the growing fatty acid chain is attached to coenzyme A. Remember, there is no coenzyme A that it is attaching to. The growing fatty acid is attached to the acyl protein carrier, the SH group of the acyl protein carrier protein. Okay. Okay. We have only 4 questions left. We will be able to finish it today quickly only. Okay, but any question in the previous this question in doubt, so let me know. Else we can move to the next question, which is in one cycle of beta oxidation, which is basically the first round, which of these enzymes are taking part? And it's very easy. We've already established this multiple times now. So you have acyl coenzyme A dehydrogenase, enoyl coenzyme A hydratase, beta hydroxyacyl coenzyme A dehydrogenase, acetyl coenzyme A carboxylase, and beta ketothiolase, which of course, as you have already guessed, absolutely correct. A, B, C, E. D is involved. D is ACC, acetyl coenzyme A carboxylase, which is involved in fatty acid synthesis. The question is about oxidation. Fantastic. Okay, next one. Now, this one says in an experiment, you are inhibiting ATP citrate lyase. Citrate lyase or ATP citrate lyase, which is same. So, if there is ATP citrate lyase inhibition, what will be the consequence? So, I told you what citrate lyase does. So, if citrate lyase is not acting, what will not happen and what can it lead to? So let me tell you again what citrate lyase does. Acetyl coenzyme A moves from the mitochondria to the cytosol via combining with oxaloacetate to form citrate. So acetyl coenzyme A plus oxaloacetate in the mitochondria forms citrate. This citrate moves to the cytosol. In the cytosol, citrate is acted upon by ATP citrate lyase, which gives it again into the two forms, acetyl coenzyme A and oxaloacetate. So you have these two products which are formed in the cytosol, acetyl coenzyme A and oxaloacetate. Now think if this activity is stopped, what effect can it do? Okay. A and C are correct, but at the same time, you have B and E as well, right? So see, if there is less acetyl coenzyme A that we have already established, and there is accumulation of citrate because you will not have citrate breakdown. Now, if you don't have acetyl coenzyme A, and we know acetyl coenzyme A is responsible for beginning of fatty acid synthesis with malonyl coenzyme A. So if you don't have acetyl coenzyme A, your fatty acid synthesis will not take place. And we have decreased NADPH production from malic enzyme because again, you don't have NADPH there, right? So you have, because NADPH is, as we know, if you go to fatty acid synthesis, wait a minute. Yeah, you can see in fatty acid synthesis, you have the usage of NADPH. This is not done anymore. So there is decreased rate of use. Yes, production of malic enzyme because there is no breaking of NADPH is happening, right? So these are all there. What doesn't happen is your increased malonyl A formation because that will not happen. Because if citrate lyase is inhibited, acetyl coenzyme A level drop, there is no malonyl coenzyme A formation because you don't have We remember how malonyl coenzyme is formed, right? We have seen bicarbonate incorporation and which gives rise to the malonyl coenzyme. So if you don't have enough this thing, enough acetyl coenzyme, there will be no malonyl coenzyme formation. So you do get this type of question which requires you to think, join all the dots. So I thought this will be a good exercise for you to revise. And I think we are on the last two questions, which will complete very quickly. So it says a cell has high level of NADPH. Again, same kind of question. Cell has high level of NADPH, acetyl coenzyme, and citrate in the cytosol. So what could be possible outcome here? So you can see it from the perspective of which metabolites are present. You have NADPH, you have acetyl coenzyme A, and you have citrate. And these are all part of different pathways, like TCA is there, your fatty acid synthesis is there. And you can connect all of these pathways. You can see which one is happening. You can also go back a step and see, okay, for this synthesis, this step is important. So this is also going to be there. So this is like a food for thought, and you can think a lot of on these lines if you read other pathways. Okay, A and D, A correct, D correct. We also have B, which is correct, right? We'll see why. First of all, let's see the question. The question says you have high level of NADPH, acetyl coenzyme, citrate. So if you have high level of NADPH and you have high level of acetyl coenzyme A, it means that you have high these lipogenic conditions, right? Lipogens is basically you are making lipogens. So you have high lipogenic conditions. Okay. Now, citrate, if you have high in the cytosol, it means that there is some problem in the TCA, because citrate should also be incorporated into TCA and that should be used up, right? But it is not being used up. So there is some problem in TCA. And the next one says that your citrate, if we know, because citrate is there, citrate positively influences the acetyl coenzyme carboxylase activity, right? So which increases the malonyl coenzyme A content. So based on that deduction, if you look into the answer, which says fatty acid synthesis is likely at life, which is lipogenic conditions. Second says the TCA cycle is inhibited at isocitrate dehydrogenase. Exactly what is the problem here? If you have more of citrate and it is not being utilized, it means TCA is somewhere not working properly, right? Fourth says malonyl coenzyme A activity levels are likely high. That's because you have citrate, which will activate ACC. And you remember, if you have ACC, which is high, then you have the acetyl coenzyme A conversion to malonyl coenzyme A, right? Now, what is incorrect is acetyl coenzyme A carboxylate is phosphorylated. It is incorrect because if acetyl coenzyme A is phosphorylated, it gets deactivated, right? So it is uh, so it is not deactive, it is activated, right? And last one says fatty acid oxidation is likely active, which is incorrect because we have established it is synthesis which is more likely to take place. Okay, all right. Last one, which is again a very, very simple question. It just says that which of these are intermediates of cholesterol biosynthesis. We haven't discussed cholesterol biosynthesis, but in case it is, I thought I'll put it as a question just for you to know which all these are part of cholesterol biosynthesis. Any idea which one is the correct answers, or which one is not part of cholesterol biosynthesis, or which one is part of cholesterol biosynthesis? Any guesses? So basically, of course, we haven't discussed, but when you are looking into lipid biosynthesis, cholesterol biosynthesis is also a part of it. It's a very important part of it. So that's why I put up this question just in case you ever revise, or if you are, when you're revising, you have this question as your revision point. Okay, maybe a minute more and then we can just wrap up today's session. Yes, sure. That's what I'm saying. No problem. So all of these are part except for the last one, which is citrate is not part of it. So if you look into cholesterol biosynthesis pathway, you will see that it begins with acetyl coenzyme A. Then it forms this HMG coenzyme A. Then it forms the malonic acid. Then it forms something called isopentyl pyrophosphate IPP. Then it forms choline. And at the end, it forms lenosterol, which forms the cholesterol. So that's the whole pathway. Citrate, however, it's not really part of these cholesterol biosynthesis. Citrate, as you know, it plays a role in the transport of acetyl coenzyme A, but it is not part of cholesterol biosynthesis. So rest everything is part of cholesterol biosynthesis in the manner, in the order that it is given, except for you have a IPP here and you have lenosterol here. Okay. All right. So if you have any question for today's session or anything in general, you can put up in the chat, or otherwise we are done with today's session. So thank you so much. We'll see each other on next Tuesday. Thank you so much. Thank you.